Great. So today I'm going to be talking about a method for trying to extract those synaptic coupling strengths. And the overall concept of this course, I don't know if you guys got a chance to look at the README, but there's kind of three conceptual steps. First, we're going to start off with our base data as just having spike times from a bunch of different neurons in our data set. And we're going to look to see when do we see more spike-spike synchronies than we would expect by chance, given that we have this labeled data set that I mentioned during the early talk, where we know that this level of synchrony usually corresponds to a true synaptic connection. Once we have these in our pocket, then what we need to do is we need to see how those synaptic strengths vary over time. And then we're going to see, all right, which ones of those pairs tend to be correlated with one another, and can we find some lower dimensional space that explains the overall correlation structure between those time series. And so I didn't know how much experience people are going to have with extracellular recording, but there's been a number of talks that people have given throughout this week, and so I can kind of breeze by this, I think. But I do want to say a little bit about the extracellular spike. And so one of the ways that we figure out which signal is coming from what source is the shape of the waveform that we record over multiple different channels. And so the spike that we're recording extracellularly, you can think about this as the inverse of what's happening inside of the cell. The positively charged ion is leaving the extracellular space and inside of the neuron. And so we record a hyperpolarization while the neuron is becoming depolarized. So different kinds of ion channels and different kinds of neuron geometries can actually give us different kinds of distributions of that waveform over physical space and different kinds of rise times and decay times to the actual shape of the waveform itself. And so using these kinds of techniques, we can figure out which neuron we're recording from, because presumably that signal should be bigger, and even some information about the kind of neuron. In particular, the excitatory and the inhibitory neurons have very different shaped waveforms, and that's actually because of some of these voltage-gated channels that are inside of the interneuron. And so all of this kind of pre-processing I've done already for you. I've already extracted these spikes from the raw data. I've assigned which spike should go along with which putative neuron, and I've made this categorization between which of those neurons I think is excitatory and inhibitory. In your own data, you're going to have your own kind of heuristic for what waveforms you think look like excitatory cells and which look like inhibitory cells. And typically, people use things like the spike width. It's actually not a solved problem at all. Now we finally have Cree driver lines where we can label all inhibitory neurons. We can give a little light ping, see who fires, and then look to see what their waveforms look like and actually figure out who are all of the inhibitory cells in a database. That's ongoing work in the lab. But this is, I would say that we can be confident that all of the narrow waveforms are inhibitory cells. We cannot be confident that all of the broad waveforms are excitatory cells. But we know that 30% of the neurons, 70% of the neurons are excitatory. So our noise rate inside of this excitatory population is going to be smaller than it could be, let's say. OK, and so these are the kinds of signals that you would record if you had tetrodes. So here, every row is data from a single unit. And you can see that the shape of the waveform varies across the different electrodes of the tetrodes. Here, there's 4 different electrodes. And you can just think about it like the microphone is closer to me than it is to you. And so my voice is going to be louder on that signal. And just to give you a sense of the kind of units that we're dealing with here, the average signal that we're recording is about 222 microvolts. A huge spiking signal would be a millivolt. So obviously, this is massively amplified for us to be able to record anything about this. And maybe you've seen these kinds of plots before. This is now plotting the amplitude of each of the spikes recorded on the first electrode and the fourth electrode. And you can see that if there's a signal that's coming from a reliable source, it ought to look the same over and over again. And so that's what these clusters are. And each of these clusters corresponds to a different neuron. And one of the tricks that we do is we can look at this and whiten the data, look at it in principal component space, and now you can really see that there's these different clusters. And so this is just how all this pre-processing has been done before you got here, and it's saved in data variable that you have. But I just wanted to give you a feeling for what does the raw data look like and the transforms we do on the data before you're looking at them. And so what are the kind of tools that are out there? Maybe you've heard about some of these things. There's different kinds of visualization softwares for manually circling these dots. Our eyes are very good at estimating spike density. And so actually during my PhD, all of those spike times were extracted by me circling those clusters manually. There's different automated and semi-automated algorithms that kind of try to estimate what the density is and model these kinds of shapes as multi-dimension Gaussians and fit the covariance matrices and estimate the center of those Gaussians and try to estimate how many different Gaussians you have. And there's other template-based methods where you say, I'm going to try to figure out what should the waveform look like on average across all of my different channels. Now I'm going to have this idealized template and go search in the raw data for when I have that template match, mark that time point, subtract that signal, and then go look to see if there's anything else happening. And the benefit of doing these template-style analyses over the clustering-style analysis is, imagine if you have two spikes that are happening at the same time. Let's say these two neurons are firing at once. What you're going to get is a summation of the two signals. So you're going to get some weird-looking waveform that's actually not really you're not going to have a cluster somewhere. It's going to be somewhere out here. And so when you're doing this kind of cluster-based analysis, those are going to be dropped. And so we call this spike shadowing. And what it means is that you can't actually resolve synchronous activity. And in parts of the brain where cells are generally silent and then they fire all at once, you can actually be losing a lot of your signal by virtue of the spikes happening at the same time. And so this is one reason to template kind of spike sorting, if you care about synchronous signals, might be the better kind of spike sorting algorithm. And so this is just an example from Kilosort. And so the real trace that we're recording now, this is a silicon probe. These are different electrodes. They're organized anatomically from dorsal to ventral. And you can see the same signals recorded on multiple different channels. And we can try to find an idealized template for what does black trace look like. And as I mentioned, then we're going to go search for that template in the raw data. And so these are now two spikes from two different units that I extracted using Kilosort. And look what happens when they happen at once. You see how the red spike is adding on top of the yellow spike? So I'm able to actually resolve those two different time points. While if I was sorting with one of these cluster-based methods, I might have the yellow spike, but then the red would be totally absent at that time point. And so if you're trying to estimate how different neurons are interacting with one another, you'd never be able to get this within millisecond level synchrony. And it's actually not even perfect with these template-based methods either. OK, so I want you now to start playing around with some of the data that I've saved and given to you. I've got, as I mentioned before, there's a bunch of files which are written in C, which you will need to compile. If you're running a Mac, which many of you are, take a look in so if you open up this directory, I don't know how it's saved, you see that I've got this file here that says compiled mex. If you've compiled the code on your computer, this file will be generated. And so if you're running a Mac, I didn't compile the Mac code, so delete that file. If you are running a Linux or PC, then try just leaving it as is. And then run that first block of code in the Synapsonable demo script. That's not a good sound. And so when I say run that first bit of code, what I mean is, if you're clicking in the script here, if you press Control-Enter, or I don't know what the Windows equivalent of that is, that should now go through and compile all of the C code and begin to load some of the important things into your workspace. And please, somebody raise your hand if you're able to do that. So some of the people running Macs. And has anyone actually received an error message? What does it say? No Mex compiler. No Mex compiler. So you need a friend. Because, yeah, so some of the, for whatever reason, you need to, with the Macs, you need to install a separate Mex compiler. But these Mex compilers are very useful. In general, one of the big limitations of MATLAB, it's slow, but if you write some things in C, you can get some very big speed increases. OK, so
do you think it's worth somebody who has compiled the Mex files in the Mac sharing them so that others can try it? Or
how many people don't have a Mex compiler? OK, so yeah, we can try.
okay,
It's not a guarantee, as you were saying, it's not a guarantee that it's going to be able to run. Does everyone who doesn't have a Max compiler sitting close to someone who has a working workspace? We've got an orphan. Anyone willing to partner up with our orphan over here? Yeah, it depends on the version of MATLAB and their operating system.
Right, you're dropping into each other. That should be so big. But the version of OS and the hardware of Mac.
So those of you who are able to load that spike struct into your workspace, go just type the spikes in and look to see what's inside of that variable. And so hopefully you can see a lot of interesting information, including what the waveforms look like, the time series. And I want you to just run this line of code here just to begin to visualize what the waveforms look like. And so hopefully you'll see that this image will pop up on your screen. Do people have that? And so these are the average waveforms on one of the channels for 2 of the different neurons in our session. And you can see how different they look. And so this is an example of the pyramidal cell is in red and the postsynaptic interneuron is in black. OK, and so as I mentioned,
Sorry, one quick thing. Can I just get a quick survey? Is anybody whose computer internet was working yesterday actually working today? No. Okay. That's all I'm going to say. Thanks.
so now we're going to begin to look to see how these 2 neurons are interacting with one another. And so as I mentioned, a very simple tool for looking at this interaction is just looking at the cross correlation. We can see here is this just discrete convolution between the 2 spike trains. And so let's run that code here. And one of the C codes that I gave you is a CCG function. And so if you weren't able to compile that, this should crash. But if you were able to compile that, why don't you run this block of code here and tell me if you see the cross correlation function appear? Yes?
Yes, just so I follow, these 2 neurons are labeled using that clustering method you just described?
Yes. So I've already gone and labeled them as a pyramidal cell and an interneuron based off of the shapes of these waveforms and their firing rate. And so in your own data set, depending on which part of the brain you're in, you're going to have to make your own decision about how you want to do those categorizations. And I'm telling you that the field does not have good ground truth data. That's still being actively collected.
Is CCG going to be different from just doing cross correlation?
It's much, much faster. Yeah. OK, so hopefully people have this image on their computer screen right now. And so you can already begin to get a lot of information about the way these pairs of neurons are interacting just by looking at this simple plot. So you see this slowly time-varying baseline. What do people think that is? I've got labeled here network co-modulation. What does that mean? LFP. So for example, both of these neurons could be firing at the same phase of theta. So in general, their spikes are happening near the same time, which is giving us this kind of envelope. What else could cause it? So phase amplitude is coupling of other kinds of rhythms. What if they're 2 different place fields and they happen to overlap? You might expect to see this kind of co-modulation too, but at slower timescales. And so I've got this overall picture of where different rhythms in the brain come from, which is that the more of the neurons that are contributing to the rhythm, the slower the frequency and the larger the brain area that's going to cover. The synaptic interaction is just 2 cells. There should be a very high frequency signal to give you a little boost above the overall co-network modulation. And we expect that this kind of synchrony should happen at a particular time lag or a particular phase. And so the overall algorithm that we wrote is tapping into both of these kind of theoretical concepts, that the synchrony should be high frequency and it should be at a particular time lag. So I say spike shadowing here. So who can tell me where this dip is coming from? I can't measure them, right? So for whatever reason, these waveforms were similar enough that I wasn't able to properly figure out which signal came from what source when they were exactly overlapping with one another. OK, so sometimes in the scripts, I have like a UI wait or something. So if you're frozen on this, then maybe you'll need to go into the command window and press Control-C. I don't remember what I did for this case. OK, so now we're basically at the point where I was in the talk, where people had been looking at these kind of peaks since like 1967 and had this dream of being able to extract what is the overall synaptic connectivity just by looking at the correlations. But we didn't really have the tools to do the right kind of perturbation experiments to extract some kind of real causal statement from the correlation. And it happened that in this case, we were generally right. But I think in looking at excitatory connections, more work is going to need to be done. All right, so we talked about this. And so the next part of this tutorial is how do we begin to assess the reliability of the spiking above and beyond that slow co-modulation. And of course, we need to define what does slow mean. And so this is like a free parameter in our analysis. And a free parameter that we played with when we were looking at our synapse detection is what do we want to consider to be synaptic timescale and what do we want to consider to be network timescale. There's no way to know this unless you have a labeled data set. And so one of the things that people had done originally to get this kind of slow variation is to jitter the spike times. Who can tell me what it means to jitter? Yeah?
You're moving the spikes around in time.
Yes, with some random time. And so the idea between this kind of shuffle distribution is you're going to break all of the high frequency synchronies and preserve the low frequency synchronies that are existing within this kind of delta t. And so this kind of jitter, where you're taking the time around the observed spike, is called spike center jitter, and it's known to have some kind of statistical problems. But it's also known to be equivalent to just convolving the cross correlation with a Gaussian. And so the jitter itself is very computationally intensive. This convolution is instant. And so this was an attractive kind of framework for me to go and do my synapse detection because you can have millions of pairwise interactions that you want to screen. And so in the demo that I show you, we're using this kind of convolution technique to estimate what should be the expected level of synchrony given this spike center jitter. But the spike center jitter and the convolution both have the same problem. So now let's say you have two totally independent spike trains, and you're going to jitter the spikes around one of them, and you're going to ask in your jitter distribution, how many synchronies do you observe above some number? And what you see is that this is not a uniform distribution. If these were totally random, and you're doing this kind of jitter, you would expect that these probabilities should be totally flat. But what you actually see is that your jitter observation, you're much more likely to see synchronies, even though the two different processes that are generating those spikes are independent of one another. So this is a conservative test. Because if you have an observed level of synchrony and you're comparing against this null distribution is going to be much higher than you would expect. So how can we begin to solve this problem and make a non-biased test? And so there's one hacky solution. And you can think of it, maybe one of the problems with this kind of null distribution is that you're already taking information that you've observed and then permuting around that observation that you've made. So you observe where the spike is already, and then you jitter around that time moment. And so it's not actually a random distribution. You're already conditioning on something that's existing in the data set. So maybe we could discount our prior observations a little bit. And so what Aaron Stark did is he said, all right, the way I'm going to discount that observation in this convolution land is let's just take a little notch out of the middle. And then we're going to convolve our convolution and look to see what do we expect to see in terms of the synchronies with our convolution in our null case versus the actual case for two independent processes. And now you get an unbiased test. But this only works, it's only non-biased and valid test if you have two independent and identically distributed variables. If you have any kind of structure in either one of them, this is still a biased test. And it's biased for the same reason that you're including some information already in your null by shuffling around your existing observation. And so for all of you people who really want to do this kind of careful destruction of the existing relationships between pairs of point processes, the right thing to do is to partition your database randomly and then jitter within those random intervals. And it turns out that this actually gives you a valid and non-biased test. And if you want to read about the proof for why that's the case, you can look at this paper here. But as I mentioned to you, that's not what I did. And the reason why I didn't do that is because I was not interested in the degree of synchrony above chance. I was interested in what is the probability of being a synapse given an observation of synchrony. And we've got a label database with which we can now begin to calibrate our thresholds.
So if I can just add one little observation, like it might seem to you like these are really arcane, sort of deep in the weeds issues, and in some sense they are, but it turns out that potentially really crucial and exciting claims like, can rats pre-play a trajectory that they've never experienced? They hinge on getting this sort of thing right. So once you really start analyzing like structure and spike trains and whether something is or is not different from chance, you really have to be careful with this sort of thing. So it's really nice that these references are up here for some cutting-edge statistical work on that.
And so I just want to give you a sense, at least with the tools that I've given you. You know the mistakes that were made, and you know that we're aware of them and kind of how to at least theoretically and conceptually fix them. But I think that if you had unlimited computational power and you were redoing it, you would do this kind of interval jitter. So how do we define our thresholds? So this is the cross-correlation function that I showed you during the talk and earlier. And we've got these confidence bounds here. With these confidence bounds, I'm saying, all right, given that we've got some kind of baseline rate function, what's the likelihood that we're going to observe some spike count above and beyond that, assuming that this is a Poisson variable? And once you formulate it like that, actually you can just use the known probability density function of a Poisson variable with this rate function to say what's the likelihood of getting X number of spikes above and beyond that. And so if you look in your code, there's a function called a BZ monosynconv click. And if you look down on line 234, you can see exactly where that calculation is being done. And basically, we're saying, all right, well, what's the likelihood of observing 0 spikes to 1 less than the number of spikes that I've observed? -1. And that's the probability of observing the number of spikes and above. And so we've got 2 different baselines that we're interested in. As I mentioned, we're interested in the slow baseline that we calculate through this convolution technique. And so this is telling us how much high-frequency synchrony we have. But we're also interested in the level of synchrony that we observe in the causal direction above and beyond the anti-causal direction. And so basically, that's going to be comparing the peak at the synaptic time lag versus all of the data that comes in before the presynaptic cell spiked. So you might imagine that if these 2 neurons were engaged in a high-frequency oscillatory dance, where one fires, the other fires, one fires, the other fires, it's going to be very difficult to establish any kind of causation there. But you're going to see high-frequency synchrony both before the presynaptic spike and after the presynaptic spike. So this comparison turns out to be very important. And as I mentioned, we have this label data set, which anybody is welcome to. This is online. And what we can do is we can say, all right, well, what's the p-value, which is really important for finding a synapse? The p-value both in the high-frequency domain, but also in the causal domain. And we can play around with these different p-values, and we can look to see which of those pairs are truly tagged synapses versus which of the pairs are not, shown in black. And then we can do what's called an Rlc analysis, a receiver operating characteristic analysis. And with each of these thresholds, we can say, what's our rate of true positives? How often do we say it's a synapse, and it was really a synapse, as determined by I can stimulate the presynaptic cell and I get a postsynaptic cell, and how many false positive rates that I have? And obviously, if I have more and more of a permissive threshold, as I keep on dropping this threshold closer and closer to 0, my true positive rate is going to be perfect. But I'm also going to have tons of false positives. And a perfect classifier would basically look like a step function. And this is getting pretty good. And we can determine what are the sets of thresholds that give us a tolerable level of true positive and false positive rates. And depending on your kind of question now, you don't need to be asking what's the level of synchrony that I observe above chance. You can be asking what's the level of true positive versus false positive rate I'm willing to accept for the kind of question I'm asking about synapses and that kind of thing.
Sorry, just to unpack that. So the juxtacellular is without the octo? Or
Yes, that's right. Those are separate experiments.
And that's the reason that's sort of like higher?
There's more data. There's much more data in the optogenetic data set.
I see. So actually the optogenetic is a better estimate because of having more data?
They're different. With the optogenetic stimulation, you're stimulating multiple neurons at once. And so you're actually driving an increased synchrony amongst a convergent presynaptic population. And so you can actually detect weak synapses because you're able to get the increase in the postsynaptic firing because they're being driven by multiple sources.
And is this on the same data or in different data sets?
Different data sets. Yeah.
But you could potentially do it on the same.
I analyze, and I did here. Yeah.
Okay. And the weak synapses, is the juxtacellular different within them than the strong synapses?
Yeah, so you're not going to be able to detect the weak synapses so well with the juxtacellular stimulation, but with the optogenetic stimulation, you can begin to reveal some of the weak synapses because you have more convergent synchrony. Yeah, so if I fire, you should do something after me, not before me. Yeah, but it's like a baseline in a critical window. Because if we're really in this oscillatory dance, the baseline just beforehand could be very important.
So it could be just correlation because it's not directional. You can't establish a direction.
We are establishing a direction between the two with the lag.
But if they're oscillating between each other?
And that's why it's so important to consider this difference.
Between the two, I see.
well, it's going to be, you need to be much higher in the positive direction than the negative direction. None of this is establishing causality. This is all a correlative method that's benchmarked around a perturbation experiment. Okay, and so the next thing to do is to now, if let's say we're recording 100 different neurons in our session, what we need to do is 100 by 100 pairwise cross correlations, and then look to see what our baseline rate of synchrony is, look to see what our confidence bounds are around the baseline, and pick which of the pairs that we have a reasonably good chance that they're going to be connected to one another. And so hopefully you can now run this line of code, and maybe this will take 30 seconds to run on your computer. But I'm telling you, if you were using Jitter, this would take you, you'd have to run this on your cluster and then come back in a couple of hours. And so as I mentioned to you, what I've already done is assigned which of these neurons is pyramidal and which is interneuron. You can get a lot of high frequency synchrony that's not synaptic. So pairs of interneurons will show very high frequency synchrony with one another, even sometimes with phase lags. And one of the things that we argued in our paper is that the source of that synchrony is actually because two different postsynaptic neurons are receiving the same presynaptic input. And so if me as a pyramidal cell is driving two of my neighbors, there's one spike that's going to make them fire at the same time. And we found that the degree of high frequency 0 millisecond synchrony that you observe between two interneurons is strongly correlated with the number of presynaptic partners they have. And if we inject current into one of those postsynaptic interneurons, you destroy that synchrony. So it's not like a gap junction or something. Another way that you can see high frequency synchrony is, let's say that I'm firing a train of spikes as a pyramidal cell as a burst. The shape of my action potential will actually change over the course of the burst. It starts, the first spike is very big, and then gets smaller and smaller and smaller. Those later spikes in the burst tend to be misappropriated to some of the other units in your data set. And so it's very common when you're looking at these cross correlations between pyramidal cells is you'll see a sharp peak at around 5 milliseconds. That's because you've misassigned the later spikes in the burst from one cell, usually with a big waveform, into the unit with a smaller waveform. And so for this reason, you're going to have a lot of false positives in my synapse detection algorithm between pairs of interneurons and between pairs of pyramidal cells. But if you precondition the data to only look for the pyramidal cells, which I label as 1 here, and the interneurons, which I label as some number higher than 1, then you'll see that most of these actually do a pretty good job, at least when we benchmark against the labeled data set. Who here is not, that script is still running to find all of the cells in the connection? Okay, but everyone else is basically run, so that's good. Okay, so now let's loop through each one of these pairs as a sanity check to see what they look like. And so here, this is one of those cases where you should see an image pop up. Just click on the image, and that'll automatically close the image and let the next image pop up. And once you get bored of looking at those cross correlations, then you can go back in your command window and Control-C out. If you want to look at all 70 or something, then you can just click 70 times and look at all of them. But this is a very important sanity check to make sure that all of this kind of computation that was just happening in the background is giving you meaningful results. And I encourage you that any kind of complicated analysis you do, make sure that you build in your pipeline these kind of visualization steps along the way to make sure you're not just analyzing noise or something, or pairs of pyramidal cells where your spike sorting was bad or something like that. Yeah, and so the variance is like if you have a very small bin, you're going to have some that are high and some that are low. And so looking at the anti-correlation gets at that a little bit, but you're right. The algorithm could definitely be improved. Yeah, it's super noisy. So, but if you're looking at this peak synchrony above what came right before, you know, that's going to be very low. And if it's a low rate in general, then the probability of getting a couple of spikes with a low rate is high, and so you're not going to be above this low varying baseline. So that's another way of taking into account. We take the absolute maximum in the anti-correlation direction. Yeah, within the same amount of time. But as I say, like the algorithm can be improved. And there's actually, this is a good example of open data where we collected this database. There's already been 3 publications with people who have built better synapse detectors on the benchmarking against the same data, but they're much slower. So if you have 1,000 neurons that you're recording, you're looking at 1,000 by 1,000 pairwise interaction, maybe you want to start with your screen doing it this quick and dirty way, and then go back and have the more statistically reasoned way later on. Ida?
The number of presynaptic connections and
Yes. Yeah.
And you can see it in this.
You can begin to look at this. So one of the things that we were interested in is when you see heterogeneity in the patterns of the pyramidal to interneuron connectivity map, is it because you're looking at two different interneuron types, for example? We know there's a huge diversity of interneuron types. And so what we did is we had a Cree driver line that just gave us axoaxonic cells. So one specific kind of interneuron. And see, those are the interneurons whose axons synapse on the axons of other cells. We see even diversity in this Cree line, and that diversity can be explained by whether or not, well, it correlates with the pattern of presynaptic input, whether those axoaxonic cells are receiving inputs from the deeper superficial neighboring pyramidal cells.
Which would correlate with their frequency.
And that's consistent. Yeah, exactly. No. OK, so maybe you guys are bored at looking at the looping through these cross correlations. But the next important thing, and this is what I was talking about at the end of my talk, is how stable is this synaptic interaction over time? And so now run this next block of code, and what this is doing is it's sub-selecting different epochs of time for doing this cross-correlation analysis. So I think the first 2,000 seconds and one is the first 2,000 seconds, and then the other one is looking at from 6,000 to 8,000 seconds. And hopefully what you see for the first example is it looks like there's some difference between the two, at least in the raw cross correlations, where during the early period, the coupling strength looks very high, and then that gets weaker for whatever reason later on. But when you do your baseline correction, you can see that actually this pairwise interaction was relatively stable. But for the other cases that I gave you, there were some dynamics there, where it looked like in the raw case there was some kind of difference, and even after you do the baseline subtraction, the difference survives. So how do we pick what's the right epoch to look at, and how do we have a time-resolved way to look to see how these coupling strengths change? And so that's going to be now the point of the next phase of the demo. OK. And so I don't know if people have looked at the README document, but the overall approach that I took with Roman Husar, who's in the back, is to try to take a generalized linear model where we're going to be predicting what's the likelihood of seeing a postsynaptic spike given two different factors. And one of those factors is what is the firing rate of that postsynaptic spike evolving over a slow time scale. And the other one is we assume that there's going to be a transient boost in that synaptic firing just when the presynaptic cell is active. And so the way to read this is this lambda function. This is going to be our intensity function for the Poisson process. This is going to be the firing rate of the postsynaptic cell. And it's going to be a function of this coarsened postsynaptic spike train. So what's the average firing rate of that postsynaptic cell over some longer time scale, some non-synaptic time scale, plus some boost that we're getting at the time of the presynaptic spike, and we're going to allow that boost to change over time. And so how do we want to allow it to change over time? You could imagine that you could model it as going exponentially stronger, or a sinusoid, or who knows what that shape is going to look like. And so we wanted to have a very flexible function. And so what we wanted to do is just say, all right, let's have some new basis set for time, which is just going to be splines that we lay out evenly over the course of the entire recording session. And so every moment in time is going to get weighted onto this new basis set. And so that's what's showing here. This is going to be our basis set, which is just going to be evenly tiling time. And this alpha function here, this is how we're going to weight each of those presynaptic spikes onto those temporal bases. This alpha is what we're going to try to learn. And that's a vector. And that's a vector that's the same length as the number of bases that we pick. And so how do we know how many bases do we want? How fast do we think that this process is evolving?
What kind of basis is it?
I'm showing you. OK. So this is the part of the code that does that. So this takes a little bit of time to do this model fitting. I think there's like 70 different pairs in the session that I gave you. I've run them all already just by running this block of code. Maybe after or some point when you feel like you know what I'm going to say, you can try running it just to see if it runs on your computer. But hold off on that for the time being. But I just want to show you where that's done. In principle, if you were doing this kind of analysis, this is the perfect place to parallelize your data, because every pair is independent of one another. So you'll take this kind of block of code, you'll put it onto the supercomputer at your university, and every node will deal with a different pair. You save the outputs, and then you go back and stitch it all back together again. OK. And so what kind of free parameters do we have in this analysis? Of course, one of the biggest free parameters is what do we consider to be a slow baseline firing rate for the postsynaptic cell. And here we're going to be very conservative. We're going to consider that the postsynaptic firing rate is that's slow, is changing on the order of 15 milliseconds. So this should handle things like firing at the same phase of theta, maybe even being involved in the same ripples, certainly firing in the same receptive fields. And if you want to look in this part of the code and follow along, this block of code is inside of this fit plasticity version 3. So you can open up that bit of code and follow along. And so the first thing we're going to do is just bin the postsynaptic spike train at 15 milliseconds. And then we're going to try to predict the firing of the cell on an 8 millisecond basis. And so we're going to do linear interpolate the bin firing rate back up again. And so if you're looking at that README document, these two lines of code, these two equations correspond to this block of code here. OK, and so what do I mean by the basis set? This is our basis set. So this is how we're going to be describing time. And so what I'm showing you here on the x-axis is our basis, and on the y-axis is time. And you can see that these neighboring epochs of time here get loaded onto different bases. And so, of course, our spikes are not going to be perfectly distributed in time like this. So you can think about this as different ways that we want to be lumping the spikes together. And so in this first example, every spike doesn't quite get treated independently. If I'm trying to learn how much I consider the first 50 spikes, they're all going to be assigned one number, one alpha function that I'm going to be fitting in our GLM. And then the next 50 spikes are going to be corresponding to the next basis. And so that's going to have its own number that's going to be fit when we do our model fitting. If we have fewer bases, that means now the first 500 spikes are going to be treated as one entity. And so we're going to see how strongly those spikes are driving the postsynaptic cell. There's going to be kind of one number that's going to be describing those first 500 spikes. And so what we're learning here is a vector that we want to be applying to each of these different bases. And we're going to try a couple of different time scales, and then through cross-validation, see which one best explains the data. Roman, why is it a cubic spline? You can see here, this is the shape.
So you could, even if you did like a Fourier basis set or something like that, you would get like different speeds of dynamic. Or no, is that good?
Yep, you could do that too.
Yep. Then you would get multiple dynamical scales.
Yeah, you could definitely do that too. OK, and so where does the magic happen? So the magic with these kinds of models happens in how you define your cost function. And so in this case, this is going to be in a function that's called NLL rate. And so that's a negative log likelihood. And so the negative log likelihood is the comparison against the model that you're defining versus the observed spike train. And so I forget if this block of code is in, I think it should be in the NLL rate function. You should see this block of code. And what we're doing is first we're going to define how the presynaptic cell should couple to the postsynaptic cell. So we're going to be trying to fit our set of weights, which are going to be multiplied by our basis. And so this term here is going to be our presynaptic contribution. And we're going to add that onto this function ops.rate. That's the course and postsynaptic firing rate, which is pre-computed and passed in. And so what we're going to do now is try all different kinds of permutations of BTA to try to see when do we predict there to be a high postsynaptic firing rate at the time of observing a spike. And then get the likelihood of that function, and then try to minimize the negative log likelihood of that function. And then keep in our pocket the set of coupling weights that best do that. Yes?
Is that really the sum of all the presynaptic weights? How is that? Is that for everything that's coming in?
What do you mean?
That pre up there.
Yes.
How many things go into that?
It's the whole presynaptic spike train.
Okay.
Yeah, one cell.
One cell.
Yeah,
Okay.
one cell. It's all pairwise.
Oh, I see.
Yeah. And so the idea is the contribution of the other cells is going to get sucked into this, the quiescent rate. That's your network. Okay. And so this is all what I've said. And if you're interested into where this negative log likelihood function comes from, you can look at some of the work by Emery Brown or Uri Eden, who've really done a lot of work into kind of formalizing this Poisson point process framework and to do the kind of model fitting. But this is where the equation is coming from. And we could have like a whole other hour seminar about deriving these kinds of equations. Yes, I'll put it on the Slack. I'll put a bunch of papers on the Slack.
There's a really nice textbook by Uri Eden and colleagues that tutorial style intros with code for this.
Yeah.
We'll post that one too.
Yeah, yeah, thank you. Okay, so this concept of cross-validation, who here has come across that before? Is somebody willing to just describe to the class what I mean by that? Or what cross-validation is? Yeah?
It's parsing your data in a way where you have test, training, and validation sets.
And what do you mean, what does that mean?
Well, you want to assess your algorithm in one part of your data and then apply that. You want to apply it. So if we're maximizing, we're trying to find the time scale, right? We find a good time scale for our test data and then apply that same, for our training data and then apply that same time scale to our test data to ensure that it generalizes.
Exactly, and I think the key part is that the training and the test data are not the same data.
Yes.
And so the risk is that if I was using every spike as its own basis set, and then building the same model that to train the GLM as I was testing it on, I could just perfectly predict spike by spike how that function should change. But if you ever saw another spike, then you would be totally overfitting the data and you would have no ability to generalize. And so this is a nice way to have some generalizability, is to do train with one set, test on another, and then it actually doesn't matter how many different model parameters you have, because obviously if you're having more different bases, you're going to be fitting more different parameters, which is also going to trivially get you better fits. And so this is, we do a 50-50 split in our data here, where we take, I think, every other block of time. Is that how you did it, Roman?
Yes, so essentially it's every other 100, so it's segmented into 100-second splits. And then you take all the odd 100-second segments, and that's what you train on. And then you take the even ones, and that's what you test on. So it's like an even out split of the data, like a true-false cross-validation. Where you do like a 50-50 split, but you just do it digitally because you're trying to fit something that's time resolved.
Yeah. Yeah. And so once you have the good fit, now we can actually predict what we want our postsynaptic firing rate to be every time there's a presynaptic spike. And so in the same function here, you can see that's our lambda full. And we have a kind of our null model, which is what do we expect the postsynaptic firing rate to be if we never consider the presynaptic cell at all. And that's what we're going to call our baseline. And the difference between the two, you can think of that's the contribution of the presynaptic cell of interest. And so now we're going to have that variable at every time that the presynaptic cell is firing. And then what we want to do is to have that time at every moment in our session so that we can begin to time lock that with other events of interest. So we can just convolve that value and normalize by the presynaptic rate. And if you look in the function, the Poisson fit, that's what these lines of code are doing. And what you're left at the end is now this measure of how much of a boost in the postsynaptic firing rate do you get given the presynaptic input above and beyond the slowly varying time scale. And what's nice about operating in this generalized linear model land is now let's say that you know that there's other things that should be affecting the postsynaptic firing rate. You can put that into your model as well and regress out some of that variance too. So for example, ripples are high frequency. So you could put the ripple phase in here. We know that the presynaptic drive is changing as a function of the interspike interval of the presynaptic cell because of short-term plasticity. If I fire twice in a row, in general, my second spike in the train is going to be less potent than the first. So you can model some of those short-term plasticity dynamics as well in the same framework. Okay, so that's basically our whole framework for estimating the synaptic coupling over time. And as I mentioned, in general, this takes maybe a minute per pair. And so you would parallelize that, run them all, and then bring them all back together again. And so hopefully you've got a directory that says processed, where there's a different file for each one of those pairs. And so let's load all of those now back into your workspace. And that's what this next block of code is doing. And let's look at a couple of them. Do people have this image on their computer? So somebody tell me, what do they see? What kind of patterns do you see here? Yeah, it's super variable, right? Like what looks like it could be a very, very effective synapse at one moment, looks like it's basically functionally inactive at another moment. What else do people see? These are 4 different examples. Yeah?
It kind of looks like they're anti-correlated across the.
Some of them, right?
Yeah.
Like these two.
Yeah.
And these two, right? Oh, and these two look like they could be correlated with one another. So it looks like that maybe there's some, not every synapse is acting independently of one another. You know, maybe there's some latent variable that's explaining how these different pairs are covariant. And that's the next part of this demo, is trying to figure out, is there some lower dimensional space with which we can represent this data to try to lump together those pairs that are covarying with one another. And so maybe people have seen these kinds of assembly analyses done on the firing rates. This is an assembly style analysis developed in part by this Lopes de Santos. I guess he was a graduate student working with Adriana Tort. But we're applying this now to the synaptic dynamics, and so we're going to call these synapse ensembles, which was a word, I think, coined by Yuri Bujaki. And so you can see in this main synapse ensemble demo, this line of code here is what's doing that kind of detection. OK, and so our first trick is going to be we're going to try to do this independent component analysis on a reduced dimensionality space. So the first thing we're going to do is we're going to project these time series onto the first n principal components. And one of the reasons why we do that is that, of course, we're going to have some measurement noise. And so our measurement noise, if we're doing independent component analysis, we're going to be more likely to ascribe each one of those pairs as being an independent thing because of that measurement noise. But by projecting the data onto a lower dimensional space, we can try to kind of filter out some of that noise and be looking at just the things that are covarying across the different pairs. And so how do we pick what's the right number of principal components to look at? Well, it turns out some mathematicians have looked at this issue and have actually modeled what would we expect our eigenvalues to look like if we had pairs of independently distributed variables. And so given that we have independent variables, what's the number of the independent variables we have, how much data do we have, then we can actually calculate what's the distribution of eigenvalues you would expect. And then you can have a hard cutoff and say, OK, well, I'll take my first n eigenvalues that are bigger than that. And the rest of those are probably due to noise. There's other kinds of techniques for getting that threshold. For example, you could shuffle your data in different ways. But in the demo that I sent you, that's the technique that we chose. And so if you're looking at some of the publications for what that distribution looks like, and for what that the eigenvalue threshold is, you might notice that there's a standard deviation term here, but we're going to z-score our data. And so if you notice that there's a mismatch in our code between what this equation is and what it says in the script, that's the reason why we don't need to worry about the standard deviation. OK. And so the next thing we're going to do is independent component analysis. Who here has run independent component analysis before? Is somebody willing to explain what's the overall thinking behind it? What about the concept, if not the algorithm?
It clusters things with similarities.
Yeah, it's similar variance. That's like the end result, for sure. I think the overall concept is you're trying to look to find some kind of latent variables which are fluctuating independently of one another. And you're going to try to map on your observed data onto those latent variables to find the best possible mapping, which is going to give you those independent fluctuations. And so what does an independent fluctuation look like? Well, it turns out that if you take a bunch of random variables and you add them together, you always get a normal distribution. And so one of the ways to look to see if you have a bunch of a case where you don't have a bunch of random variables is to look for something that's non-normal. And so you can look at how Gaussianly distributed your latent process is and use that as your cost function to try to find what are the best weights to go from your original data onto your independent component. And so if you're looking at this fast ICA script, you can see that that's where this magic is coming from. Where you've got this description here of what does it mean to be Gaussian distributed, and you're actually going to be doing your parameter fits to try to find what's the best beta weights to go onto the n number of independent components as we described before from the expectation of that n from chance. Okay, I don't know how clear that was. So do people have questions? Which part? Yes. So you want to try to find a latent variable that's non-Gaussian. Because the more Gaussian you are, the more likely you're going to be the sum of a bunch of random variables. That's the overall logic behind this the fitting function for the independent component analysis. But you can imagine that there could be other explicit tests for independence. And those would be kind of different algorithms for doing the independent component analysis. So unlike PCA, there's not one solution for the ICA. There's going to be different algorithms to get you to the overall conclusion, which is you want to cluster together the data that covariates.
But for ICA, you really need to assume that people know that the data is not Gaussian, because otherwise.
Yep, that's true.
Check before we start.
It's not, but it's true. If you have a bunch of variables that are themselves distributed in a Gaussian way, then if you keep on adding up Gaussian variables, then you're going to get a Gaussian variable. I'm sure that people have thought about that. I don't know if there's some aficionados in the crowd who've done independent component analysis who've come across that before. But that's like a good hyperparameter to consider in the nature of your data. That like the choice of algorithm for your independent component analysis is going to depend on the distribution of your data in the first place. Okay, and so I think now we can go back to the script, and I didn't put here which line of code to run, but I have it in the comments. So hopefully you can see that after you try running that assembly detection, and then we can start plotting all of the data, but now sorted by our assembly weights. And what you're looking at here is every row is a pair, every column is time, and the color axis is the synaptic coupling strength of that pair at that time. And now you can really see, I cherry-picked 4 examples for you, but you can see this huge amount of structure that exists in the entire ensemble, where it looks like that there's something that's happening here that's making these pairs go up at the expense of these other pairs. So these are anti-correlated with one another. And if you look at our independent components, you can see that actually we need 14 different components to explain most of the variance in this data set here. And hopefully you can try to match up which of these components corresponds to which of the overall patterns. Like maybe this block here is corresponding to this block, and you see it reactivates over here, and it kind of reactivates over here. I've tried to sort these somehow to preserve where I think of the pairs that are contributing. For example, this block here kind of looks like that. And now I can correlate each one of those pairs time series, the synaptic coupling strengths with each other, but I can sort this correlation matrix according to how they're weighting onto the independent component analysis. You can really see this kind of correlation structure here with different kinds of synapses that seem to be part of the same kind of ensemble being driven by the same latent process, which we don't know what it is. I mean, now we've got a new signal, a new neuroscience signal to look for to see what's driving these groups to be firing together. The X's, I just wanted to see if you have the same presynaptic cell, the same presynaptic partner. Where are you on this matrix? So each of these rows and columns, there's a presynaptic cell and a postsynaptic cell, and that some are going to have the same presynaptic, some the same postsynaptic, some will be independent. So just as a way to visualize, does the same presynaptic cell always correlate with one another? The answer is no. If you have the same postsynaptic cell you always correlated, the answer is no. It really looks like a pairwise phenomenon. And so one of the things that, of course, working in the Bujaki lab we wanted to look at is sleep and wake. So here's an example of 2 of the pairs. And here we've just coded this session as the animal awake or asleep. And you can see the transitions tend to happen around these sleep-wake states. And actually what I find super interesting is that this one that's increasing when the animal's awake also has these little blips when the animal's in REM sleep. And so we know that those are both high acetylcholine states. So maybe acetylcholine could be one of these neuromodulators, which is gating the synapse. And with this, I'd like to say thank you. This work is really born out of a nice collaboration with Roman over the last year. So thanks to him in particular, and it's the same thanks I gave earlier. So thank you for your attention.
For a couple questions, if there are any remaining.
Yeah.
Go back to the clean map over time. So I know these cells tend to be, and these aren't necessarily, these cells necessarily connections, but these cells tend to be directionally modulated on a linear track. Do you anticipate that coupling strength might change?
Yeah,
for
I do.
the right routes?
I do. If you had enough data, I bet you could show it. But you need it. I mean, to get every point estimate, you need like 5 minutes. So you would really need a lot of data with the animal running one way and a lot of data with the animal running the other. But yeah, I bet you're going to see that one of the things that's decorrelating the running direction is going to be the inhibitory drive. And actually, if you look to see how the cells fire on the linear track on the very first trials, they're not directionally dependent. They start as place fields that fire on both and then stop firing in one, which is already giving us a hint that actually that's being inhibited in some circumstances and not others. Ida.
Did you make any graphs of them and do just graph sorts of analysis? Because these are pairwise, but it's much more interesting to see who's receiving a lot more presynaptic and going to fewer cells, who's like, you know what I mean?
If there's some graph network people in the audience, I would love that kind of collaboration. I've been really trying to.
Yeah, we did that.
Great. Yeah.
So you said it was hard for this algorithm to work on excitor variability.
Yes.
Why was that?
Because you need to worry about the spike sorting problem. So you're going to get tons of false positives. One reason. The other reason is that these interneurons have a very high firing rate and sit very close to the threshold. And so any little kick you're going to fire, you don't really change the overall firing rate that much. What you're doing is just redistributing exactly in time when that spike is going to happen. There's another reason, which is that there's a single release site from the presynaptic cell to these fast spiking interneurons. So that when you're looking to see these fluctuate over time, you're not actually asking the question of like which bouton is being released. Like you're only looking at one bouton at a time and one release site at a time. Yeah. All right. Well, thank you, everybody.